So here's the chapter known as reproduction. We are doing MCAT biology. So we'll just do a review for this chapter. All mammals have certain characteristics similar, that is mammary gland, 3 bones in the middle ear, 1 lower jaw, fur, hair, heterodont, sebaceous gland. There are 2 groups which produce young ones in a very different method. First is Prototherians. These are duck-billed platypus, echidna, both produces eggs, oviparity. The another one is metatherians, koalas, kangaroos. Their fetus undergo some development in the womb and some out of the womb, which is in the pocket-like structure. Then comes the cell cycle, which is very important. Cell cycle has different phases. We will be studying each and every phase one by one. First one is the G0 phase. That G0 phase is the phase where the cell is not at all preparing for the reproduction. It is just existing. Then comes G1. In this phase, the cell is trying to create organelles for the energy and protein, mitochondria, endoplasmic reticulum, ribosomes are used. All these functions have started. Then comes the S phase, where the genetic material start to replicate. Every chromosome contains 2 identical chromatids that are bound together at a specialized region known as centromeres. Then there is the G2 phase. G2 phase is the checking phase, where the genetic material must be perfect to be transferred. Then comes M phase. M phase is the phase for mitosis and cytokinesis. Then there is a diagram for centromere, which replicate and forms 2 sister cells. In case during the checking of DNA, which is done during the G2 phase, if during the checking of the DNA it appears that the DNA is faulty, then it undergoes the repair. The main protein responsible for the repairing is P53, and which is a very important point to note because it's a bit unusual thing. Then comes when the damaged DNA passed to next stage of the division. Without correction, it causes abnormalities like cancer. Oncogenes are cancer-causing genes. There are tumor suppressor genes present to suppress this tumor. Okay, so now let's dig into the mitosis. This happens in asexual reproduction, where 2 identical daughter cells are created from a single cell. It has 2 phases. First one is cytokinesis. The second one is actually mitosis. So, firstly, the cell contains nucleus which is surrounded by nuclear membrane and have chromatin material inside it flowing freely. The chromatin material condenses into the chromosomes, nuclear membrane disappears. During the prophase, the centrioles in the cytoplasm flow towards the poles, and then the spindle fibers start to grow. These fibers get attached to the either side of the chromosome, and which forms the second stage, which is metaphase. The alignment into the straight line at the equator happens. Then comes the anaphase, in which the spindle fibers pulls the chromosome apart from each other. And then comes the telophase, where the cytokinesis occurs by the division of the cell into 2 parts. Then the nuclear membrane appears again and the chromatin material flow normally, resulting in the formation of 2 cells from one. So this is a continuous process, and dividing it into the phases is very difficult. That's why I have explained in a big paragraph form where we can sort of differentiate the phases, but majorly the beginning part comes into the prophase where the chromatin material condenses into the chromosomes. Actually, the material is into a flowy form, then it condenses inside the nucleus. As it condenses, it becomes chromosomes. As it becomes chromosome, the nuclear membranes start to disappear. Then what happens? The chromosomes start flowing. As they start flowing, the centrioles in the cytoplasm flow towards the poles. From the poles, the spindle fiber appears. Then spindle fiber catch these chromosome and make it to be aligned into the center of the cell, which is equator. Then these spindle fibers pull them apart and divide it into two parts. Then as they are pulled them apart, they go to the poles, on both the poles. Then at the middle of the cell, the breakage happens, and the cell will start to squeeze down and slowly get differentiated. I mean, separated and become two different cells. So during this process, sort of duplication of the material happens. That's why the cell or the daughter cells become identical to each other. So this was the process of mitosis. Now let's go for the meiosis. During the mitosis occurs in somatic tissue and result in two daughters which are identical. But in meiosis, in gametocyte germ cells, this process usually occurs, and this result up to 4 non-identical sex cells, which is known as gametes. Meiosis happens in two phases, which has prophase, metaphase, telophase, and a phase 1. Same as prophase, metaphase, anaphase, telophase 2. So during the prophase 1, the chromatin material convert into the chromosome pairs happen, attached to the centromere, have total of 4 chromosomes attached together. Allele exchange occurs between the 4 attached chromosomes. Then what happens? Then comes the metaphase. Same thing happens. As in meiosis, what happened? The chromosomes were attached together, and then as the nuclear membrane get disappeared, the spindle fibers appears. Same happens here. As the nuclear membrane disappear, spindle fiber rises from the centrioles. These centrioles are something which are produced after this disappearance of the nucleus membrane, and they are produced at the poles, and from these centrioles the spindle fibers arises. So as the spindle fibers arise from the centriole and attach with the chromosome each side, they again align them into the equator and are separated by the spindle fibers pulling force. As the spindle fiber pull them apart, half part of both the chromosomes, actually 4 of them are attached together, so a pair just get pulled towards the equator. After that, telophase occurs, which is same as the meiosis. The cell divide from the central part, and we have 2 different daughter cells. Because at the beginning there were 4 chromosomes attached together, and now they are separated, so The daughter cells are not identical to each other. After this, the meiosis 2 happens. In this, prophase 1 has not prophase 2 starts, in which the chromatin material again arranged as chromosomes. Then the spindle fiber arise again. Spindle fibers attach to the chromosomes, pull them apart, take them to the poles. Then again the same thing happens. There is splitting of the cells occurs, and they form another pair of daughter cells. So at the end of this process, there are 4 daughter cells which are not identical to each other. So we will study about the reproductive system in the next video.